Section number 43 of Gray's Anatomy Part 4. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by M.L. Cohen, Anatomy of the Human Body Part 4 by Henry Gray, the sympathetic nerves. The sympathetic nervous system innervates all the smooth muscles and the various glands of the body and the striated muscles of the heart. The efferent sympathetic fibers, which leave the central nervous system in connection with certain of the cranial and spinal nerves, all end in sympathetic ganglia and are known as preganglionic fibers. From these ganglia, postganglionic fibers arise and conduct impulses to the different organs. In addition, afferent or sensory fibers connect many of these structures with the central nervous system. The peripheral portion of the sympathetic nervous system is characterized by the presence of numerous ganglia and complicated plexuses. These ganglia are connected with the central nervous system by three groups of sympathetic efferent or preganglionic fibers, that is, the cranial, the thoracolumbar, and the sacral. These outflows of sympathetic fibers are separated by intervals where no connections exist. The cranial and sacral sympathetics are often grouped together owing to the resemblance between the reactions produced by stimulating them and by the effects of certain drugs. Acetylcholine, for example, when injected intravenously in very small doses, produces the same effect as the stimulation of the cranial or sacral sympathetics, while the introduction of adrenaline produces the same effect as the stimulation of the thoracolumbar sympathetics. Much of our present knowledge of the sympathetic nervous system has been acquired through the application of various drugs, especially nicotine, which paralyzes connections or synapses between the preganglionic and postganglionic fibers of the sympathetic nerves. When it is injected into the general circulation, all such synapses are paralyzed. When it is applied locally on a ganglion, only the synapses occurring in that particular ganglion are paralyzed. Langley, who has contributed greatly to our knowledge, adapted a terminology somewhat different from that used here and still different from that used by the pharmacologist. This has led to considerable confusion, as shown by the arrangement of the terms in the following columns. Gaskell has used the term involuntary nervous system. Gray, sympathetic nervous system. Langley, autonomic nervous system. Meyer and Gottlieb, vegetative nervous system. Gray, craniosacral sympathetics. Langley, parasympathetics. Meyer and Gottlieb, autonomic. Gray, oculomotor sympathetics. Langley, tectal autonomics. Meyer and Gottlieb, cranial autonomics. Gray, facial sympathetics, glossopharyngeal sympathetics, vagal sympathetics. Langley, bulbar autonomics. Gray, sacral sympathetics. Langley, sacral autonomics. Meyer and Gottlieb, sacral autonomics. Gray, thoracolumbar sympathetics. Langley, sympathetic. Meyer and Gottlieb, sympathetic. Gray, enteric. Langley, thoracic autonomic and enteric. Meyer and Gottlieb, enteric. The cranial sympathetics. The cranial sympathetics include sympathetic efferent fibers in the oculomotor, facial, glossopharyngeal, and vagus nerves, as well as sympathetic afferent in the last three nerves. The sympathetic efferent fibers of the oculomotor nerve probably arise from cells in the anterior part of the oculomotor nucleus, which is located in the tegmentum of the midbrain. These preganglionic fibers run with the third nerve into the orbit and pass to the ciliary ganglion, where they terminate by forming synapses with sympathetic motor neurons whose axons, postganglionic fibers, proceed as the short ciliary nerves to the eyeball. Here they supply motor fibers to the ciliaris muscle and the sphincter pupillae muscle. So far as known, there are no sympathetic afferent fibers connected with the nerve. The sympathetic efferent fibers of the facial nerve are supposed to arise from the small cells of the facial nucleus. According to some authors, the fibers to the salivary glands arise from a special nucleus, the superior salivatory nucleus, consisting of cells scattered in a reticular formation, dorsomedial to the facial nucleus. These preganglionic fibers are distributed partly through the chorda tympani and lingual nerve to the submaxillary ganglion, where they terminate about the cell bodies of neurons whose axons as postganglionic fibers conduct secretory and vasodilatory impulses to the submaxillary and sublingual glands. Other preganglionic fibers of the facial nerve pass via the great superficial petrosal nerve to the sphenopalatine ganglion, where they form synapses with neurons whose postganglionic fibers are distributed with the superior maxillary nerve as vasodilator and secretory fibers to the mucous membranes of the nose, soft palate, tongues, uveal, roof of the mouth, upper lips and gums, parotid, and orbital glands. There are supposed to be a few sympathetic afferent fibers connected with the facial nerve, whose cell bodies lie in the geniculate ganglion, but very little is known about them. The sympathetic afferent fibers of the glossopharyngeal nerve are supposed to arise either in the dorsal nucleus, nucleus ala cinerea, or in a distinct nucleus, the inferior salivatory nucleus situated near the dorsal nucleus. These preganglionic fibers pass into the tympanic branch of the glossopharyngeal and then with the small superficial petrosal nerve to the otic ganglion. Postganglionic fibers, vasodilator, and secretory fibers are distributed to the parotid gland, to the mucous membrane and its glands on the tongue, the floor of the mouth, and the lower gums. Sympathetic afferent fibers, whose cells of origin lie in the superior or inferior ganglion of the trunk, are supposed to terminate in the dorsal nucleus. Very little is known of the peripheral distribution of these fibers. The sympathetic efferent fibers of the vagus nerve are supposed to arise in the dorsal nucleus, nucleus ala cinerea. These preganglionic fibers are all supposed to end in sympathetic ganglia situated in or near the organs supplied by the vagus sympathetics. The inhibitory fibers to the heart probably terminate in the small ganglia of the heart wall, especially the atrium, from which inhibitory postganglionic fibers are distributed to the musculature. The preganglionic motor fibers to the esophagus, the stomach, the small intestine, and the greater part of the large intestine are supposed to terminate in the plexuses of Auerbach, from which postganglionic fibers are distributed to the smooth muscles of these organs. Other fibers pass to the smooth muscles of the bronchial tree and to the gallbladder and its ducts. In addition, the vagus is believed to contain secretory fibers to the stomach and pancreas. It probably contains many other efferent fibers than those enumerated above. Sympathetic afferent fibers of the vagus, whose cells of origin lie in the jugular ganglion or the ganglion nodosum, probably terminate in the dorsal nucleus of the medulla oblongata, or according to some authors, in the nucleus of the tractus solitarius. Peripherally, the fibers are supposed to be distributed to the various organs supplied by the sympathetic efferent nerves. The sacral sympathetics. The sacral sympathetic efferent fibers leave the spinal cord with the anterior roots of the second, third, and fourth sacral nerves. These small medullated preganglionic fibers are collected together in the pelvis into the nervus erigentes, or pelvic nerve, which proceeds to the hypogastric or pelvic plexuses, from which postganglionic fibers are distributed to the pelvic viscera. Motor fibers pass to the smooth muscle of the descending colon, rectum, anus, and bladder. Vasodilators are distributed to these organs and to the external genitalia, while inhibitory fibers probably pass to the smooth muscles of the external genitalia. Afferent sympathetic fibers conduct impulses from the pelvic viscera to the second, third, and fourth sacral nerves. Their cells of origin lie in the spinal ganglia. The thoracolumbar sympathetics. The thoracolumbar sympathetic fibers arise from the dorsolateral region of the anterior column of the gray matter of the spinal cord and pass with the anterior roots of all the thoracic and upper 2 or 3 lumbar spinal nerves. These preganglionic fibers enter the white rami communicantes and proceed to the sympathetic trunk, where many of them end in its ganglia. Others pass to the prevertebral plexuses and terminate in its collateral ganglia. The postganglionic fibers have a wide distribution. The vasoconstrictor fibers to the blood vessels of the skin of the trunk and limbs, for example, leave the spinal cord as preganglionic fibers in all the thoracic and upper 2 or 3 lumbar spinal nerves and terminate in the ganglia of the sympathetic trunk, either in the ganglion directly connected with its ramus or in neighboring ganglia. Postganglionic fibers arise in these ganglia, pass through the gray rami communicantes to all the spinal nerves, and are distributed with their cutaneous branches, ultimately leaving these branches to join the small arteries. The postganglionic fibers do not necessarily return to the same spinal nerves which contain the corresponding preganglionic fibers. The vasoconstrictor fibers to the head come from the upper thoracic nerves. The preganglionic fibers end in the superior cervical ganglion. The postganglionic fibers pass through the internal carotid nerve and branch from it to join the sensory branches of the various cranial nerves, especially the trigeminal nerve. Other fibers to the deep structures and the salivary glands probably accompany the arteries. The postganglionic vasoconstrictor fibers to the blood vessels of the abdominal viscera arise in the prevertebral or collateral ganglion, in which terminate many preganglionic fibers. Vasoconstrictor fibers to the pelvic viscera arise from the inferior mesenteric ganglia. The pilomotor fibers to the hairs and the motor fibers to the sweat glands apparently have a distribution similar to that of the vasoconstrictors of the skin. A vasoconstrictor center has been located by the physiologist in the neighborhood of the facial nucleus. Axons from its cells are supposed to descend in the spinal cord to terminate about cell bodies of the preganglionic fibers located in the dorsolateral portion of the anterior column of the thoracic and upper lumbar region. The motor supply to the dilator pupillae muscle of the eye comes from preganglionic sympathetic fibers, which leave the spinal cord with the anterior roots of the upper thoracic nerves. These fibers pass to the sympathetic trunk through the white rami communicantes and terminate in the superior cervical ganglion. Postganglionic fibers from the superior cervical ganglion pass through the internal carotid nerve and into the ophthalmic division of the trigeminal nerve to the orbit, where the long ciliary nerves conduct the impulses to the eyeball and dilator pupillae muscle. The cell bodies of these preganglionic fibers are connected with fibers which descend from the midbrain. Other postganglionic fibers from the superior cervical ganglion are distributed as secretory fibers to the salivary glands, the lacrimal glands, and to the small glands of the mucous membranes of the nose, mouth, and pharynx. The thoracic sympathetic supply accelerated nerves to the heart. They are supposed to emerge from the spinal cord in the anterior roots of the upper 4 or 5 thoracic nerves and pass with the white rami to the first thoracic ganglion. Here some terminate, others pass in the ansa subclavia to the inferior cervical ganglion. The postganglionic fibers pass from these ganglia partly through the ansa subclavia to the heart. On their way, they intermingle with sympathetic fibers from the vagus to form the cardiac plexus. Inhibitory fibers to the smooth musculature of the stomach, the small intestine, and most of the large intestine are supposed to emerge in the anterior roots of the lower thoracic and upper lumbar nerves. These fibers pass through the white rami and sympathetic trunk and are conveyed by the splanchnic nerves to the prevertebral plexus, where they terminate in the collateral ganglia. From the celiac and superior mesenteric ganglia, postganglionic fibers, inhibitory, are distributed to the stomach, the small intestine, and most of the large intestine. Inhibitory fibers to the descending colon, the rectum, and the internal sphincter ani are probably postganglionic fibers from the inferior mesenteric ganglion. The thoracolumbar sympathetics are characterized by the presence of numerous ganglia, which may be divided into two groups, central and collateral. The central ganglia are arranged in two vertical rows, one on either side of the midline, situated partly in front and partly at the sides of the vertebral column. Each ganglion is joined by intervening nervous cores to adjacent ganglia, so that two chains, the sympathetic trunks, are formed. The collateral ganglia are found in connection with the three great prevertebral plexuses placed within the thorax, abdomen, and pelvis, respectively. The sympathetic trunks, truncus sympatheticus, gangliated cord, extend from the base of the skull to the coccyx. The cephalic end of each is continued upward through the carotid canal into the skull and forms a plexus on the internal carotid artery. The caudal ends of the trunk converge and end in a single ganglion, the ganglion impar, placed in front of the coccyx. The ganglia of each trunk are distinguished as cervical, thoracic, lumbar, and sacral, and, except in the neck, they are closely correspond in number to the vertebrae. They are arranged thus: cervical portion, 3 ganglia; thoracic portion, 12 ganglia; lumbar portion, 4 ganglia; sacral portion, 4 or 5 ganglia. In the neck, the ganglia lie in front of the transverse processes of the vertebrae; in the thoracic region, in front of the heads of the ribs; in the lumbar region, on the sides of the vertebral bodies; and in the sacral region, in front of the sacrum. Connection with the spinal nerves. Communications are established between the sympathetic and spinal nerves through what are known as gray and white rami communicantes. The gray rami convey sympathetic fibers to the spinal nerves, and the white rami transmit spinal fibers to the sympathetic. Each spinal nerve receives a gray ramus communicans from the sympathetic trunk, but white rami are not supplied by all the spinal nerves. White rami are derived from the first thoracic to the first lumbar nerves inclusive, while the visceral branches which run from the second, third, and fourth sacral nerves directly to the pelvic plexus of the sympathetic belong to this category. The fibers which reach the sympathetic through the white rami communicantes are medullated. Those which spring from the cells of the sympathetic ganglia are almost entirely non-medullated. The sympathetic nerves consist of efferent and afferent fibers, the origin and course of which are described subsequently. The three great gangliated plexuses, collateral ganglia, are situated in front of the vertebral column in the thoracic, abdominal, and pelvic regions, and are named respectively the cardiac, the solar or epigastric, and the hypogastric plexuses. They consist of collection of nerves and ganglia, the nerve being derived from the sympathetic trunks and from the cerebrospinal nerves. They distribute branches to the viscera. Development. The ganglion cells of the sympathetic system are derived from the cells of the neural crests. As these crests move forward along the sides of the neural tube and become segmented off to form the spinal ganglia, certain cells detach themselves from the ventral margins of the crests and migrate towards the sides of the aorta. Some of them are grouped to form the ganglia of the sympathetic trunks, while others undergo a further migration and form the ganglia of the prevertebral and visceral plexuses. The ciliary, sphenopalatine, otic, and submaxillary ganglia, which are found on the branches of the trigeminal nerve, are formed by groups of cells which have migrated from part of the neural crest, which gives rise to the semilunar ganglion. Some of the cells of the ciliary ganglion are said to migrate from the neural tube along the oculomotor nerve. End of section 43.